Bismillahirrahmanirrahim. Welcome back, guys. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about level of regulation of gene expression. In the previous video lecture we just talked the regulation of gene expression. Okay, that how the gene expression are regulated, or we just talked that what is actually a regulation of gene expression. We just talked about that. But in this video we are going to talk about level of regulation of gene expression. Okay, level of regulation of gene expression. What does it mean? It means that when we have level of regulation of gene expression means that there are certain level through which we can control the gene expression. For example, as you can see, we have what let's suppose we have central dogma process. Let me a little bit once again tell you. In the central dogma of the cell biology, we have what? We have the DNA. Let's suppose this is the DNA. Let's suppose we have a DNA. The DNA is obtained through a replication process. Through replication, what we get? We get a mature DNA. And after the replication, we have a transcription process. Transcription. What actually happened in transcription? In transcription, we have the mRNA production. Means from transcription we get the mRNA, and from this mRNA, from transcription, then we have another process which we call that translation. And after translation, we have another process also. We call that post-translational process. Post-translational or post-translational process of the cell, of the protein, or post-translational modification as well. Now, what happened? We have replication. In replication, what we get? We get the DNA. Okay, and from DNA, transcription means the synthesis of the RNA from the DNA. Okay, so then we get what? We get the RNA synthesis, mostly mRNA, and after that we get the protein. The protein synthesis is actually called as translation. When the protein are synthesized from the mRNA, we call that translation. And after translation, another process is occur which we call that post-translation, in which the protein are in a functional, or we get functional protein. This is actually level of gene. This is what? This is level of gene expression. Means these are actually the level which the gene are expressed. From this level we get the RNA, and from this level we get the protein, and from this protein level we get the functional protein. This is the level of the gene expression. Now we are going to talk about the level of regulation of gene expression. It means that we can control, or the cell can control or regulate the expression of the genes through different level. Okay, mostly the cell can regulate the gene expression at that level and at that level. Means there are certain protein which act upon on the DNA. So when it act upon a DNA, so then the DNA will be means that gene will turn on, and when the gene is turned on, so then we get mRNA protein and functional protein as well. But still there are some certain protein which we call that inhibitor, which act upon on the genes of the genes inside the DNA, due to which the whole process will be halted. This is the first level through which we can regulate the gene of expression. Another level we call that mRNA gene regulation level. If let's suppose the mRNA is formed, so then there are certain protein which act upon on the mRNA, and when the protein are act upon on the mRNA, so then what happened? The enzyme which act upon on the mRNA, so then we didn't get the protein and functional protein. And when there are actually some sequences which we call that enhancer. Through this enhancer sequencing, the activator protein are bound, and then the gene expression will occur. there are some sequences which is occur at the downstream. It is also occur at the upstream, but most of the time it occur at the downstream of the DNA or of the promoter as well. So at the downstream, we have some sequences which we call as silencer sequences. At that sequencing, the inhibitor protein will bind and it will cause the halting of the central dogma of the cell biology, or it can stop the whole process, as you can see. And there are some protein which we call it activator, which can bind on the sequencing which is present upstream of the promoter area. We call that activator, or we call that enhancer sequencing. So this is enhancer sequence. The activator protein will bind and the process will occur, means the gene expression will turn on. Okay, so the gene expression is turned on at that area. It is also turned on off at that area by certain type of the protein and enzyme. And the gene expression is also turned on and turned off at that particular area as well. So these are actually the two level of the gene expression. These are the two level of the regulation of the gene expression. Okay, sometimes there are also most of the time this process does not occur, but sometime at that particular portion of the genetic expression at that particular area, we have also some types of particular protein and enzyme which bind on this protein portion, and it can inhibit the functional protein. Most of the time it not occur, but sometime if we get the protein, so then there are certain protein bind on it and it will cause the destruction of the protein or stop the protein. Due to this way, the functional protein we didn't get. If we didn't get the functional protein, the cell will not perform their function, or means the enzymes are not formed, which are not used for the certain purposes. This is actually the level of the gene expression. Means there are specific level through which we can express, which we can regulate the expression of the gene expression. So that's all about the level of the regulation of the gene expression. I hope this video helpful. If you like this video, make sure to hit the like button, share this video, and subscribe the channel. Inshallah, in the next video, we are going to talk about regulation of gene expression in prokaryote in a detail. Okay, so please stay tuned and keep watching. Thank you so much.